All right, guys, we are going through the different categories, structural categories of these neurotransmitters. We did acetylcholine and biogenic amines, talked a little bit about dopamine, a little bit about serotonin, and how drugs can affect their quantity within the synaptic clefts. But we have three more to do. We have amino acids as a category and peptides as a category. Amino acids and peptides are similar in that an amino acid is really tiny, right? It's a single amino acid. Remember, amino acids come together to form a protein, but an amino acid is just one single unit of that protein. So this is to say that there are neurotransmitters that small, okay? They're technically not a protein. They're just one unit, which is an amino acid. And then we also have peptides, which are basically just a tiny protein. Okay, so you could have 3, 4, 5 of these amino acids linked together to make what are called neuropeptides or the peptides category. Okay. Now, so that kind of ties them together, which is why I put them on the same slide. But this is a place where I would like you to know some specifics about whether a chemical excites or inhibits. It is not the main way we're categorizing them, right? We need to know the 5 categories and some examples within each. But in this particular slide, I'm going to underline what you absolutely will want to know about this particular one, GABA, which technically is not an amino acid, but it's real close to butyric acid, which is basically very close to an amino acid. This is the primary principal or primary inhibitory neurotransmitter in the brain. So I'd like you to know the main inhibitor in the brain. And keep in mind, again, right, if there's only stimulation in the brain, when it comes to reward centers, we'd be so high, we'd be high as a kite all the time. Okay? We'd be manic, bouncing off the walls. So it's just important that we have some controls over the firing and formation of action potentials, or we could say nerve impulses, in our brain, in our spinal cord. So that's a big one I want you to know. GABA is the main inhibitory one in the brain. The other one, absolutely, I want you to know what it does, is glutamate is the main or principal excitatory neurotransmitter in the brain. So it's almost like GABA, they're the brakes in the brain, right? They're the decelerators when it comes to formation of action potentials. And glutamate is the accelerator, right? This one makes the car go fast, this one makes it go slow, and there's a balance going on in between, okay? So that I would like you to know, okay? That these two in particular are amino acids, so they're real small, and they're the main inhibitory with GABA or excitatory neurotransmitters with glutamate when it comes to the brain. Now, I'm going to show you some examples of what these do and how they work here in a second. But before we go too far, I'm just going to add this to the slide. So chloride is actually, I don't know if it's going to come out, but chloride is actually the ion. You can't see the purple against the blue background there. Chloride is the ion that is being allowed to pass when GABA binds its receptors. Okay? And then down here with glutamate, well, that allows for calcium to pass. So let's just change that color because it's not very easy to see. So chloride is what's allowed to pass with GABA. That's why it's inhibitory. And calcium is the ion that's allowed to pass when it comes to glutamate in the brain. And that's why it's excitatory. All right, so now this other group, the peptide group, this is a group of our natural opiates, believe it or not. So you've heard of opiate drugs. You've heard of the opiate epidemic. Heroin is the one you hear a lot in the news about, but you also hear about all sorts of other opiate drugs, some of which are super, super potent, okay? And can absolutely have a big effect in the body. And of course, it's causing overdoses are causing deaths and so on. But these natural opiates, these are actually naturally occurring chemicals that we're now talking about. Endorphins and enkephalins are actually our own natural opiates. They bind receptors and have an effect, oftentimes reducing pain, okay? And in some cases, increasing euphoria, increasing your mood, and improving your mood, okay? So we have to keep in mind that the reason that these illicit drugs work, like heroin, or some of these other controlled substances that you need a prescription for, or you need to have administered within the hospital, like codeine or morphine, these kinds of opiates are not natural, but they're binding the same receptors that our natural opiates do. Okay? So some examples I've got of natural opiates when it comes to endorphins, these particular chemicals are what give you the runner's high. Okay, or when you're working out and you have the production of endorphins, that is going to kind of take away the pain, and also adds to some mood enhancing when it happens. Enkephalins, these would be our natural opiates released during labor, for example. So when a woman is going through labor, pains and contractions, giving birth, these natural opiates are released to reduce that pain. Now, I've had plenty of young mothers and in my class, and they've told me, no, that doesn't happen because they felt a good amount of pain. But believe it or not, there is still a dulling of pain in that process, perhaps not as much as people might like. Something that's interesting about the endorphins that I read years ago, there was a guy who was he was incarcerated for some period of time for use and distribution of opiates and illegal drugs, and he did his time, and he was released, and he was an amazing runner, long distance runner, and he actually kind of made a name for himself as an amazing athlete. But he reflected upon this in an interview in a magazine one time, saying that he gets just a small percentage of the feeling of what he used to get when he would take some of these drugs. In that runner's high was just a small little bit. It's because it's not nearly as potent as the high quantities of drugs, of concentration of opiates he was taking in at the time, years and years ago. Another peptide is a substance P, which is ironically a mediator of pain signals. So while endorphins, which are small peptides, dull pain and cause an increased mood, improved mood, substance P, which is also a peptide, actually increases pain signals. So, within this group, we have two diametrically opposed chemicals, one that makes you feel pain and one that dulls pain. And certainly pain plays a role, and an important role, in that when you feel pain, you tend to stop doing whatever is, or try to mediate that, whatever is causing that pain. And so it's good that we feel pain for that reason. But there are also some natural ways that we dull that pain with natural opiates. All right, so this next slide really just kind of explains GABA in a little more detail. The fact that it opens chloride channels, hyperpolarizes the cell, so it's going to have a harder time of firing. And certain drugs actually interact with GABA receptors. And so we would say that they interact in a similar way. Okay? So they bind GABA receptors, allow chloride to pass, and inhibit firing of the next cell. This whole list kind of makes sense when you think about how it is that you're inhibiting in the central nervous system. So barbiturates, benzodiazepine, Ambien or Sonata, these help you to go to sleep, right? They're going to be inhibitors. They're going to be downers in a way, right? That's the kind of the term we give this whole set of drugs. And alcohol. Now this is an interesting one because here we're saying that GABA receptors at the synapse, which decide whether, at that point, whether a cell is going to fire or not, that these receptors are being stimulated and it's going to decrease the rate of that neuron firing, okay? So it's at the GABA receptor that we're saying alcohol can behave like GABA and it can slow the firing of that cell. But alcohol also, if you recall, back in an earlier clip, alcohol also slows the conduction of a nerve impulse. So here we see now that alcohol not only decreases the tendency of forming the action potential on the cell, but also, from before, decrease the speed at which the impulse moves. So it's kind of an inhibitor, a downer, in two ways. It's inhibiting the formation of the action potential by acting like GABA does at the synapse, and it's slowing the movement of that action potential once the action potential forms. All right, so what I've done here is I put together some circuitry of some neurons communicating with one another, just to kind of illustrate how complex this can get. Okay, so here we have our dopamine cell, a dopa neuron. This is to say that it produces dopamine, and it releases that dopamine down here at the end of the axon. But this dopamine neuron is receiving signals, it's a postsynaptic cell, from a GABA neuron. Now, we know that GABA neurons, if they're producing GABA, they're going to be inhibitory. They're going to cause chloride to pass into a cell. Okay? So if we have let's just say we have GABA being released here, okay? A certain amount of GABA being released, because the GABA neuron is going to fire at a certain rate. Okay? And as those action potentials reach the end of this axon, calcium goes in, synaptic vesicles fuse, release their contents. And we know that down here on the postsynaptic cell, what we're going to find is that when GABA binds its receptor, it's going to allow chloride to pass into the cell. And chloride is a negatively charged ion that's going to cause us to hyperpolarize that cell. Now, if that's all the stimulation that dopamine neuron received, the dopamine neuron would never fire, would it? Because we're making the line go down. We're going further away from an action potential. But keep in mind that these different neurons in the brain receive signals from hundreds of other neurons, okay? So we have to keep in mind that there's a bunch of other, so I drew in a couple of other axons here, there's a bunch of other cells actually stimulating that dopamine neuron. All the GABA neuron is doing is keeping that in check to a degree, okay? So the dopamine neuron, is it going to fire? Absolutely, it's going to fire at a certain rate, just not as fast as it would without this inhibition from above. So let's just say this dopamine neuron is firing, okay, at a certain rate, and releasing dopamine down here upon a whole set of circuits called the reward center in your brain. This determines your mood. This determines how good are you feeling, okay? And so I put this corny smiley face here because it keeps your mood up, okay? The firing of those neurons in that particular part of the brain keep you feeling all right. Now, let's say that we add in another factor here, okay? There happen to be opiates, natural opiates, that are produced in the brain, okay? Here we have our GABA neuron. It's slowing the firing of the dopamine neuron, but we still have a certain stimulation of the reward center. Now let's add in what natural opiates can do. Okay, so let's switch to, we'll go to black for this one. Okay, so our opiate neuron, this is going to release a certain amount of, let's say, endorphins or enkephalins. And the way these particular chemicals work is they bind receptors that are found in a very different location. In this case, natural opiate receptors are found near the end of the axon of other neurons. In this case, the end of a GABA axon. Now, let's say we have a release of a certain amount of natural opiates. And these natural opiates bind the receptor at near the end of this GABA neuron. What is going to happen? What is the effect of this? Well, the effect of the binding of natural opiates is it decreases the rate at which the cell it's binding to releases its neurotransmitter. So we see a certain rate, 1, 2, 3, 4, a certain amount of GABA being released to inhibit the next cell, right? It's keeping this dopamine neuron in check. It's not going to get out of hand, okay? But when natural opiates are released, they slow the release of that GABA neurotransmitter. So I'm going to cross off a couple of these action potentials. So now, let's say GABA is being released at half the speed that it was before. So then I'm going to take out roughly half. Okay, we'll get rid of 3 of those. Okay, we're getting rid of roughly half of those neurotransmitters, and now they're going to bind their receptors over on the other side half as much. It's going to inhibit the dopamine neuron half as much. If this is the case, well, then dopamine is going to fire more rapidly, isn't it? You're inhibiting the inhibitor, aren't you, right? This opiate release is inhibiting the release of an inhibitor. So the next cell is going to fire more rapidly. Keep in mind, again, all sorts of excitatory neurons are telling dopamine neurons to fire. Now, as we reduce the effect of GABA, we're taking the brakes off. Okay? So what we're going to find now is that dopamine is going to fire, these neurons are going to fire more rapidly. That's going to increase the amount of dopamine released in that part of the brain. It will bind receptors in the neurons of this reward center. And guess what? You're going to feel great, right? You're going to have euphoria. You're going to feel so good. And this is why, partly why, these opiates are causing you to feel good, okay? It reduces the inhibition of a basic set of circuits that help your reward circuitry to fire. Now, ironically, opiates also play a role when it comes to the stimulation of pain, but they directly inhibit the release of substance P. So the interesting thing about opiates is they do 2 things. They decrease the inhibition on reward centers, so you're feeling up, but they also decrease directly the release of substance P, so you feel less pain. All right. I remember watching a intervention program. The name of the show is Intervention. I don't know if you guys have seen it, with my sister-in-law and my wife, and we were watching this show where there's this woman. She was buck naked on the show. She was very much under the influence of heroin. Okay, she had actually hurt herself. She was bleeding somewhere on the. I mean, not a. It wasn't a life or death situation, but she was bleeding. She had hurt herself, and she was feeling just fine. So she couldn't feel the pain, and she was euphoric at the same time. Okay. Now you can see then how it is that we need to control these kinds of drugs, right? This is why they're controlled substances, because these natural opiates, endorphins and enkephalins, do this for us. But if you greatly increase the opiates binding to this receptor with illicit drugs like heroin, for example, or I think OxyContin, and there's another one now, I'm forgetting it for some reason, that's super, it's a synthetic drug that binds the same receptors. It really is addictive. You can see why they have to be under some control. So what do I have here for you? I forget what I've got in the next. Okay, so I'm putting these out here just, I guess I had more time on my hands to show you some of these images, but again, check out where we used to have these actually advertised. So Bayer had pharmaceutical products, including aspirin, or hey, you could pick up some heroin. And it's a sedative for coughs, I guess that's what they were toting it as. That it's a miracle cure. And when I see these images, I got to share with you that buddy of mine who burned his back with Nair years ago. I was showing him how to do a procedure in the laboratory one time years ago, and the procedure required the use of a needle, a syringe and a needle. But the needle is not even the kind of needle that you would use to inject anything. It had a blunt end to it, so it'd be the last thing you want to use to actually puncture the skin and introduce a drug or something. But we were using it in a procedure in the lab, and I was showing him how to do the procedure because he had never done it, and I had. So I'm showing him these things, and so he picks up the syringe as a joke. It was just him and me. And there's this rubber tubing that was there hanging from, like you see in chemistry labs, hanging from the tap, from the water tap. And he proceeds to put it around his arm like they do to get a blood vessel to show. And he put the needle up as if he was doing that, like it was supposed to be a joke. And so I look and I kind of laughed. And what I didn't realize, what my buddy saw right when he looked up, doing this with the needle, is one of the students, because we were TAs, we were teaching labs at the time, and these students, these undergraduates would show up. We were graduate students. They would show up every now and then. The student comes around the corner and sees my friend doing that. And my buddy, he's like, oh my God, that kid saw me doing that. So he got up to go and find the guy, and they must have run away. They must have freaked out, because who knows what they thought, right? Seeing him doing that. They may not have known there was nothing in the needle, in the syringe at all. But anyway, I just thought I'd share that story with you. Okay, the last one. This is kind of a catch-all group. Okay, the fifth category is novel messengers. So kind of unique chemicals. And I'm only including one example here. So you only need to know one for me, okay, in this group. But it's kind of a unique one. This neurotransmitter doesn't fit into the other categories well. And one of the reasons it doesn't is because it is a gas. It's actually a gas molecule that's dissolved in your body. It's called nitric oxide, okay, NO for nitric oxide. And it plays a really big role with learning and memory, so it's key when it comes to learning and memory. It also plays a role with relaxing smooth muscles, like smooth muscles around blood vessels. So it plays a key role in blood pressure, okay? If it binds, it allows for the relaxation of those muscles around those pipes that are your blood vessels, and the pressure within would go down. So this is pretty important. Now, early studies with nitric oxide brought us to the understanding that, one, it was a gas, and it was also a non-polar molecule. It has lipid-like properties in that way. So this is not only a gas that's a neurotransmitter, but it goes right through the membrane and has its effect on receptors inside the cell. It's not binding to receptors on the surface of the membrane, which is another thing that makes it very potent. Like steroid hormones are potent because they go through the membrane with no problem. Now, because there was a realization that nitric oxide played a role with smooth muscles and thus blood pressure, a lot of big companies that want to make money will actually and have actually studied drugs to affect the quantity of these chemicals in your body because they want to help treat other people that have high blood pressure. High blood pressure is a real problem. Cardiovascular disease linked to high blood pressure kills a lot of people. And so a lot of scientists are hired, right, by companies that want to make a little bit of money by treating these people that need help. But it so happens that in their trials of trying some of these drugs, they found that, yes, in some cases they are adequate to help treat high blood pressure, but in certain cases they opened up the blood vessels that entered certain organs of the body, like the male organ, the penis, right? And certain of these blood vessels that would dilate and let more blood into that organ would, of course, cause erection, okay? So this was an interesting side effect, and of course, Pfizer took advantage of this, right? Pfizer actually owns the rights to the production of Viagra, and they, of course, now advertise Viagra as an erectile dysfunction drug. And it is also true that if you have high blood pressure, they may prescribe a drug similar to this to help you with your high blood pressure. And one of the side effects is that it makes it a little easier to have an erection. Okay, so it's kind of an interesting little side effect that they came across, and of course they took advantage of it, right? All right, so I'm going to leave it at that, guys. We're done with basically chapter 12 as far as I'm concerned, what I'm going to present to you. And we're going to launch right into chapters 14 and 13 in that order, with the central nervous system first. Okay?